Hi everyone. Thank you for your interest on this webinar. I am Véronique de Conto. I am a project leader in vitro pharmacology in HCS Pharma. It's my pleasure to present you the 3D cerebral cell culture in BIOMIMESYS Brain. I will present you how improve the relevance of in vitro model by including the brain microenvironment. First of all, just some word about HCS Pharma. This company has been founded in 2014. We have two activities. Historically, we are specialized in high content screening. That means we can test a molecule for pharmaceutical, food, and cosmetic industries by phenotypic screening. It is by imaging. We can make this screening thanks to a robotic platform equipped with two automated confocal microscopes, as you can see here. And we can perform this test in 2 dimensions and also in 3 dimensions models. Indeed, we have another activities. We produce and sell BIOMIMESYS matrix. We acquired this patent in 2017. BIOMIMESYS is an innovative in vivo mimicking 3D cell culture technology. And that's why we are now focused on the development of a full range of BIOMIMESYS 3D cell culture systems for various organs and therapeutic areas. These two activities have the common goal to improve the success of preclinical research. Indeed, despite a significant selections of a drug candidate during all the process of drug discovery, the failure rate in clinical trials is 90% in average, and even more for the brain, as you can see on this graph. The probability of approval in clinical trial for a drug targeting brain diseases is only 3%. This data highlight a lack of relevance of preclinical models used upstream to select drug candidates. Indeed, animals present interspecies differences with human, and human in vitro models are often too simple because they don't take into account the microenvironment, in particular the extracellular matrix. Indeed, as you can see here, in vitro models are often in 2 dimensions with a single cell line monolayer growing on plastic, whereas in the human body, the cells grow in 3 dimensions embedded in the extracellular matrix. But what is the extracellular matrix? The matrix is composed of several molecules, including hyaluronic acid, which is a non-sulfated glycosaminoglycan in green on this graph. There are also sulfated glycosaminoglycan like heparan sulfate and chondroitin sulfate, which can bind to a core protein to form proteoglycans. There are also glycoproteins like tenascin, adhesion molecules like fibronectin, and also structural molecules like collagen. Despite this general composition, the composition of the matrix vary according to the organ, as you can see on this picture. The stiffness varies also according to the organ, as you can see on this graph. And you can see that the elastic modulus of the brain is about 0.1 kilopascal, which is very far to the rigidity of plastic commonly used for cell culture. The matrix stiffness can also change in disease, for example, in case of a brain tumor. The extracellular matrix is involved not only in cell adhesion and migration, but also in the cell differentiation and in cell proliferation through biochemical signal retention and mediation, and also through mechanical forces. The matrix is also involved in cell communication with interactions with receptors. All these characteristics make the matrix a key element in the development and in physiological functions of the human body. Moreover, the matrix can be modified and involved in a pathological mechanism. In the aim to include the extracellular matrix in vitro models, we developed BIOMIMESYS matrix. BIOMIMESYS combine both the advantages of solid scaffolds, in particular the porosity and structural maintenance, and the advantages of hydrogels, in particular biotic properties and cell matrix interactions. That's why we use the term of hydroscaffold. BIOMIMESYS is a hyaluronic acid-based matrix graphed with structural and adhesions molecules. It allows to reproduce the matrix like decellularized organs, but without the difficulty of access and the problem of reproducibility. BIOMIMESYS is therefore physiological because hyaluronic acid is a major component of the extracellular matrix, chemically defined, adapted for screening, and also BIOMIMESYS can be adapted to each organ or disease because we can modulate the composition and the T-cells. In this way, we developed versions of BIOMIMESYS adapted to culture cerebral cells, which is BIOMIMESYS Brain. BIOMIMESYS Brain is composed of hyaluronic acid, RGDS, which is the cell adhesion fragment of fibronectin, collagen 4, and a cationic polymer to promote neuric outgrowth. The elastic modulus of BIOMIMESYS Brain is 0.1 kilopascal, like the brain in vivo. Just some words about the process. To produce BIOMIMESYS, first we produce an hydrogels with hyaluronic acid, adhesion proteins, and also structural proteins. But this hydrogel is not compatible and not stable for cell culture. And thanks to a patented process, we can obtain this hydroscaffold, which mimic the extracellular matrix and which is adapted to in vitro cell culture. BIOMIMESYS hydroscaffold reproduces extracellular matrix with all physicochemical and biological properties. It is the first organ-specific extracellular matrix. If we compare BIOMIMESYS to other in vitro systems, like 2D models and also other 3D models, and also with decellularized organs, we can observe that BIOMIMESYS combine all the advantages, including the organization of cells in spheroids or organoids. BIOMIMESYS is physiological and does allow cell matrix interactions. As I said, we can modulate the composition and the thickness. So BIOMIMESYS is organ specific and allow also stiffness modulation. Moreover, BIOMIMESYS is ready to use and compatible for screening. That's why BIOMIMESYS hydroscaffold is a unique technology based on biofunctionalized hyaluronic acid that is reproducible, scalable, and versatile. I will now present you some technical aspects about BIOMIMESYS and how to use BIOMIMESYS. BIOMIMESYS is provided on a lyophilized form directly cast in multi-well plates. The main tip to know for the cell seeding is to concentrate the cells in one drop of about 25 microliter in the aim to hydrate the lyophilized matrix and to catch the cells inside. After that, you can add the medium for cell culture. You can, of course, perform manual seeding, but BIOMIMESYS is also adapted for automatic seeding. An important thing to know is that the cell density has to be higher in BIOMIMESYS Brain compared to 2D cell density because of the 3D volume. As you can see on this video, the media exchange in BIOMIMESYS is quite similar to the culture because the hydroscaffold loosely adhere on the bottom of the wells and the cells are retained inside. You just need to be gentle with the pump. Interestingly, it is possible to transfer the hydroscaffold. You can take the hydroscaffold with a clamp and transfer in another vessel, as you can see on the video. It could be interesting, for example, if you want to fix only one time point. BIOMIMESYS is compatible with main downstream analytical technologies. Indeed, because BIOMIMESYS is translucent, it is adapted for microscopy and spectrometry. Because BIOMIMESYS is porous, it is adapted for PCR, Western blot, and ELISA assay. It is biodegradable, thus it is adapted for flow cytometry, and it is solid, so you can use it for histological techniques and also for in vivo implantation. I will now present you some example of results using BIOMIMESYS Brain. And first example of a neurotoxicity assessment. For neurotoxicity assessment, we can use SHSY5 cell line. This cell line is commonly used to assess neurotoxicity, both in academic research and in the pharmaceutical industry. As you can see on this picture, there are more than 150 publications per year mentioning both SHSY5 and neurotoxicity. These cells form spheroids in BIOMIMESYS Brain and can be maintained in culture during about 10 days. As you can see on this picture, with in blue the nuclei of the cells, which form spheroids, in green the beta-3 tubulin, which is a marker of neurons, and in red the dead cells. This cell line comes from neuroblastoma and can be differentiated into neurons by several methods. In this study, we tested the main differentiation methods found in the literature, in particular retinoic acid, staurosporine, and B21 plus CMP, which is a serum-free medium. We analyzed the expressions of neuronal markers, including beta-3 tubulin, synaptophysin, and tyrosine hydroxylase. And as you can see on this graph, staurosporine and B21 plus CMP medium allow to increase the expressions of the neuronal markers compared to in proliferation medium. And the neuronal maturity was higher in B21 plus CMP medium in blue on the graph. We exposed these cells culture in B21 plus CMP medium to compounds of non-toxicity. We culture the cells in 2D for comparison and in 3D in BIOMIMESYS Brain. We tested compounds known to be non-toxic, like L-ascorbic acid, toxic but non-neurotoxic, like acetaminophen, and also compounds known to be neurotoxic, including tamoxifen and cis-hydroxydopamine. We can observe that the molecule categories were respected with no toxic effect for non-neurotoxic molecules and a toxic effect for neurotoxic molecules. We can also observe that the cells are less sensitive when cultivated in BIOMIMESYS brain, in red on the graph, than when cultivated in 2 dimensions, in black on the graph. We can also use BIOMIMESYS for pharmacology assessment. For example, Parkinson's disease affects more than 10 million people worldwide. This disease is characterized mainly by movement symptoms like bradykinesia, tremor, and rigidity. These movement symptoms are caused by the death of dopaminergic neurons in the substantia nigra pars compacta on the influence of environmental and genetic factors. This neuronal death involves the extracellular matrix. There are modifications in the extracellular matrix composition with an increase in some components like collagens, and also a degradation of hyaluronic acid. In parallel, there are an increase in the hyaluronic acid receptor CD44. The extracellular matrix is also involved in the formation of Lewy bodies, which is an important marker of Parkinson's disease. And the matrix is also involved in the neuroinflammation found in neurodegenerative diseases, with the activation of glial cells by tenascin C and chondroitin sulfate proteoglycan, and also the increase of MMP lead the degradation of the extracellular matrix and the formation of bioactive fragments, which has a pro-inflammatory effect. To obtain an in vitro model of Parkinson's disease, we used Lumes cells, which are dopaminergic neuronal cell line. Here you have the Lumes cells culture in 2 dimensions for comparison. And here you have the Lumes cells culture in BIOMIMESYS brain. You can observe this picture is a higher magnification, and we can observe that the cells form clusters within in blue the nuclei with neurites stained in green for better tubulin. On this video, you can observe the Lumes cells at several Z-stacks, several levels, separated by 2 micrometers to visualize the 3D organization of the cells in BIOMIMESYS brain. We can observe that the differentiated Lumes cells in BIOMIMESYS brain express NEN, which is a marker of neuronal maturity, and also tyrosine hydroxylase, which is a marker of dopaminergic neurons. We can induce Parkinson's disease phenotypes by using some pharmacological inducers like rotenone, cis-hydroxydopamine, paracrat, glutamate, MPP+, and erastine. The exposition of Lumes cells to these 6 Parkinson's disease inducers showed that the Lumes cells are less sensitive when cultivated in 3D in BIOMIMESYS brain than when cultivated in 2D. In 2D in black on the graph, and in 3D in red on the graph. For all the tested molecules, the difference was particularly pronounced for erastine. This difference in cells sensitivity can be explained by the retentions of molecules in the matrix, like in vivo, and also by differences in protein expressions in BIOMIMESYS brain compared to 2D culture. As I said, BIOMIMESYS brain and an elastic modulus of 0.1 kilopascal, like the brain, and the Lumes cells form a cluster with neurites inside. But if we increase the stiffness of BIOMIMESYS to 1 kilopascal, for example, like in brain tumor, we can observe that the Lummes cells form compact spheroids and no more cluster with neurites. The matrix elasticity influences the cell organization. I explained that the matrix is involved in the neurodegeneration, but the glial cells are involved too. In particular, glial cells are involved in the neuroinflammation found in neurodegenerative diseases. That's why, in the aim to develop a more relevant and more complex in vitro model, we perform a co-culture between astrocytes and dopaminergic neurons in BIOMIMESYS Brain. We perform the co-culture in 2 times. First, we see the astrocytes, and 2 days later, we see the Lummes cells. You can see on the picture the astrocytes stained in green for GFAP, and the Lummes cells stained in red for beta-acetylcholine. And you can observe here the co-culture with both the astrocytes in green and the neuronal cells in red. BIOMIMESYS Brain allows sequential co-culture protocols. To summarize, BIOMIMESYS is a unique hydroscaffold reproducing the extracellular matrix. It is the only system reproducing the stiffness naturally observed in the various healthy or pathological tissues and organs, like the brain. It is composed of purified, well-controlled components, allowing batch-to-batch consequency. It is suitable for various cell culture vessels, like plates, biochip of different formats, and it is compatible with HS platforms. To conclude, BIOMIMESYS fills the gap between 3D cell culture system and in vivo for a higher predictability of your cerebral models by reproducing its extracellular matrix. For your information, we have other versions of BIOMIMESYS, including BIOMIMESYS adipose tissue, BIOMIMESYS liver, and also pathological versions of BIOMIMESYS, which is a BIOMIMESYS oncology. The plates are available in 96 well plates and also in 384 well plates for BIOMIMESYS liver and oncology. Moreover, the plates are supplied in ready-to-use format. We can also produce a BIOMIMESYS custom if you want, for example, a specific composition or a specific stiffness for your studies, or also if you want a specific vessel format, including biochips. I thank you for your attention, and do not hesitate if we have questions. You can directly ask your questions on the chat, or you can also send an email to this email address. Thank you.